So thank you to the organizers for inviting me, and thank you to all the people who survived the selection pressure that was imposed on you during the week and made it to the last session. So I'm going to talk about a mathematical model of the way a tumor and the immune system interact and dynamically, and in particular, how as a tumor grows, it can trigger the immune system response, and as the immune system responds, it can cause the tumor to regress. And so there can, if the balance is right, there can be an oscillation that ensues, despite the fact that the overall volume of the tumor might be growing. And so that sets up an inherent periodicity that is this interaction dynamic between the tumor growth and decay and the immune system. And my key point in this presentation that I really want you to focus on is that synchronizing and optimizing the chemotherapy and the immunotherapy dosing schedules with the underlying periodicity of the cancer immunity cycle can compensate for lower total dose. Okay, so that is sort of the key, that was our key hypothesis in developing this model, and so we're testing it out and developing it further. So I'm going to spend most of my time, actually, which goes, what do I have, until 2:40 or 2:30?
2:40 and plus 10 minutes for questions.
Okay. So I'm going to spend a lot of the time talking about the model because I'm going to divide this into 3 parts because the model's a little complicated. But first, what I want to do is I want to focus on what the cancer immunity cycle is. And this was not something developed by our group at all. This was something developed by Chen and Melman in 2013, and then revised just 3 years ago. And it's a super highly cited paper, very influential. Lots of clinical trials associated with it. I'll mention some of them as we go. It's a great framework for understanding how the tumor and the immune system interact. And so I want to start out by walking you through the 7 steps that they highlighted of this cycle, the basic biological mechanisms of it, okay? So there's the beginning 2 steps. I'm not going to focus on all the details of these steps so much, but I do want to break up this cycle into 3 different sort of clusters. So there's the beginning, which is the tumor growth phase. That as the tumor grows, the cancer cells release antigens, and that they present themselves to the immune system. And then there's the immune response phase, which are several steps, I think 4 steps in their cycle. Priming and activation of the T cell population, trafficking of T cells to the tumor, infiltration of T cells into the tumor, and then recognition of cancer cells by the T cells. So that would be what I would say would be, generally speaking, the immune response phase. And then there is the tumor regression phase, which would be recognition of cancer, which would be the killing of the cancer cells by the immune system. All right? And so that's obviously, idealized to some extent, but it's useful. And it's, as I say, there's hundreds of papers in the last couple of years on this topic. But amazingly, there hasn't really been a mathematical model that I know of that focuses on this cancer immunity cycle particularly, and also uses that cycle in order to try to optimize chemo and immunotherapies. All right, so let me now just extract some of the main players in this model that I'm going to be talking about. So it's going to be an evolutionary game theory model at its heart, but then there's going to be other features to it. And basically, it's a compartmental model as opposed to a finite cell model. But you can view these compartmental models as the expectation value of some finite population of cells taking place in the competition. Either way is fine. But basically, think of this as a compartmental deterministic model. And then what we do with this model is we actually then run it stochastically by letting the parameters in the model vary stochastically. So in the end, that allows us to do a uncertainty quantification, characterization, something called the Sobel analysis, which is a variance-based method. So if I have time, I'm going to show you just a little bit of that, how to interpret this kind of a model stochastically that way. But anyway, let me just describe the model. So there's three sort of groups of players in this game. There's the T cell population, they're the cancer cell population, and they're the healthy cell population. And they're interacting with each other, mediated by the tumor microenvironment, which functions as, you can think of it as kind of an immunostat, which kind of, for the immune system. And then with this cycle going through, our goal would be to use two different control functions, one for the chemotherapy, which applies selection pressure directly on the cancer cells, and immunotherapy, a separate controller, which applies indirect pressure on the cancer cells by changing the activation threshold of the immune system. Okay, so those are two mechanisms by which our model is going to be influencing this cycle. And so keep these things in mind. There's going to be a fundamental period of the cancer immunity cycle, and I typically call that PCIC. And then we're going to try to dose synchronize with that cycle. Yeah.
Just one brief question.
Sure.
Would you assume that the healthy cells are also attacked by the chemotherapy, or are they?
No. In this model, they're not. So basically, the cancer cells are the ones that are sensitive to the chemotherapy because their proliferation rate is so high compared to the healthy cells. So they're just more.
You don't have proliferation in the model, so to say.
In this kind of a model, we don't. I could certainly include more compartments and have sensitive and resistant cell populations and things like that, but it just gets more complicated.
Quick question.
Yeah.
What is the interaction between the healthy cells and the T cells? Is there an arrow going from the healthy cells to the T cells?
So I used to think of this as a rock, paper, scissors kind of a game, which is why I constructed this figure, but the rock, paper, scissors is a red herring. This is not a rock, paper, scissors game. So I'm just sort of indicating that these arrows form a cycle. And let's just put it this way, that the healthy cells, in a certain way, if there were no cancer cells, and you're just a healthy guy sitting there, your healthy cells are going to keep your immune system in check. So your T cells, in some sense, if you think of it in terms of a rock, paper, scissors game, would be a rock over a scissor. But don't keep that.
Yeah, no, sure, yeah.
And I have a small remark related to the question that was just asked. When you say chemotherapy, do you use traditional chemotherapy, or could this include small molecule inhibitors, which specifically target cancer cells and leave the healthy cells alone?
You'll see what it is in our model. It basically is applying selection pressure on the cancer cells. So I'm not particularly modeling one kind of chemotherapy over another.
To answer your question, so for such therapies, they don't really affect healthy cells.
No, they don't really affect healthy cells.
That would be good. Yeah.
Yes.
It's consistent with your.
Yes, they don't really affect healthy cells. Okay, so this is. Wait, what happens here? All right, so this is part one. This is the first of basically 3 slides that explains the model. So the basic players are X1, X2, and N. That's the healthy cells, the cancer cells, and the T cells, let's say. And the way the dynamics works is that the healthy cells and the tumor cells are playing a replicator dynamics evolutionary game with the fitness. F1 is the fitness of the healthy cells, F2 is the fitness of the tumor cells, and bracket F is the average fitness of the whole group. And I will have a lot to say about how these fitness functions are constructed in part 2 of the model. So for now, just know that, of course, these are not constants. These are functions of the frequencies X1 and X2, and the system is a cubic nonlinear interacting problem, okay, for the replicator equations. There's the T cell population, which is the N equation, and the guts of it is a logistic kind of a nonlinearity, but the key is that there's a term here, which is an activation or an inhibition term. It could be positive or negative, all right? So it could, the immune system, the T cell population can grow and it can decay based on the sign of H. And H is going to be a function of the cancer cell distribution. And theta of T is going to be my immunotherapy control knob, all right? And then there's going to be, then there's this term S here, which is a, let's see, let me show it all because I don't like this. So the S term is a T cell suppression mechanism that increases with tumor burden and adjusts for how immunogenic the tumor is. All right, so I've written it first as S of V of T. V of T is the volume of the tumor. So it's going to be a function, if you look down here, it's a function that is larger for larger tumor volumes. And it also has a denominator which has a function in it which indicates how immunogenic the tumor is, meaning how sensitive the tumor is to the immune system. And that changes in time. Typically, larger tumors are less immunogenic than smaller tumors.
Why don't you have V equals x1 plus x2?
Why don't I what?
Why don't you just say that V is equal to x1 plus x2, an algebraic relation?
V?
V, the tumor size, equals, equals to x2.
Oh, right. I don't think that's a great model, but it is a model. I mean, keep in mind, x1 plus x2 equals 1. So
I didn't mean to say that. I meant to say x2. V should be just x2, right?
yes. You could say it's a function of x2. Put it that way, and that's what this is going to be. I'll show you what that is in a minute. That is a model. I'm not saying that wouldn't work. It's a little, yeah. You'll see in a second what we do. So as you point out, and as I'm saying, as I'll show you in a minute, this growth rate, alpha G, is a function of time and a function of x1 and x2. So it will be a function of the fitness variables. And I'll show you that on the next slide. There's obviously a tumor carrying capacity. And this I of T is the immunogenicity function that models immune evasion under selection pressure. And then there's a parameter epsilon, which we can control, which is the immune response timescale. Basically tells you how quickly the immune system responds to changes in the tumor size. And then there's delta, which I think of as a discrepancy parameter, which is actually a constant, and you're going to see how that affects things. Basically, as the tumor goes through one cycle, it's going to grow at a certain rate, let's say, and then the immune system kicks in, and the T cell population then causes some regression of the tumor, and the growth is faster and more powerful than the regression. So over one period, there's a discrepancy there. If the discrepancy weren't there, the immune system itself in one cycle would basically kill the tumor, which is not realistic. Yeah?
I'm a little confused also about X2 and V. And particularly if you look at your T cells, why is your expansion in terms of the T cells proportional to X2 rather than to V? If V is the tumor volume, right?
Yes, V is a tumor volume. You're confused about this term?
No, about up the H of X2.
Well, I'm going to show that on the next page.
Because you have the expansion term of the tumor proportional to X2, but your tumor is actually V. Why is it not proportional to V?
Should be Vx2 then, maybe H of V times x2.
Well, let me go through the next slide, and then you can revisit those questions.
Okay.
Yeah.
Is there a question here? So, if there's no tumor, does it somehow arise in this model? Because I would expect that if x2 is 0 or V is 0, then it...
If x2 is 0, then x1 is 1, and it's all healthy cells.
Oh, okay, and then your V equations just kind of start from 0. As soon as you have a little bit of tumor, then your dynamic kicks in.
Yeah. Okay, so this is an important point here. There's an activation threshold, which is important, and that is 1 over 1 plus theta, where theta is my immunotherapy control. And what that means is that if the cancer cell population is below this activation threshold, then H is positive, which means the immune system kicks in. And if the cancer cell population is above this threshold, then, then H is positive and the immune system kicks in. So if it's below the threshold, the immune system decays. And if it's above the threshold, the immune system kicks in. And you can vary this by varying theta. Okay, so you can lower that threshold by varying theta. And that is our immunotherapy action. All right? It's going to be a function of time, but that's how we're going to impose immunotherapy. So it indirectly puts pressure on the cancer cells by lowering the activation threshold. All right. So as I said, theta of t is our immunotherapy control function. C of t is going to be our chemotherapy control function. As I said, it lowers the activation threshold, so that would be like checkpoint blockade immunotherapy. And C of t imposes selection pressure directly on the cancer cells. And then, as a standard, the healthy plus cancer cell population is 1, so it's frequency. And the volume is between 0 and k, and we've normalized things, so n is between 0 and 1. All right? So that's the basic framework of the model. But there's a lot more, particularly this, how we define the fitnesses.
When does CFT appear?
It doesn't yet, but it will. It will in the next, when I start, part 3 of the model, I'm going to tell you all about theta of t and C of t.
Thank you.
Okay, part 2, all about the fitness, the replicator equations, and how we build the payoff matrix associated with replicator equations, because this is totally crucial to the way the model works. So you're probably, I don't know, some of you, I know at least one of you is familiar, no, many of you are familiar with the replicator equations. So that's the replicator equation system, x1 and x2. A is our 2x2 payoff matrix. It's a cubic nonlinear equation. That's the average fitness. That is the fitness of population I, which would be either the healthy cells or the cancer cells. And the key to this whole thing is how we define the payoff matrix, all right? And just for a second, imagine the following. The entries of this payoff matrix, typically, it's a 2x2 matrix in this case. The entries are going to be functions of the immune system, n of t. So they're time dependent, and they are functions of n. But we do it in a certain way. So I think of n in this model as the environment, which is changing the payoff matrix entries. And here's how it changes it. So we take a growth payoff matrix, which is basically a prisoner's dilemma matrix, that if there was nothing else, the evolutionary game would just saturate to 1. That's in the tumor growth phase. And then we have a regression payoff matrix, which is a prisoner's dilemma matrix with the rows switched. So if there were nothing else, then the tumor would regress. And it doesn't regress at the same rate as it grows because we have different baseline values, although both are prisoner's dilemma payoff matrices, this and that, all right? But what's key is that we take a convex combination of these two matrices through this function, 1 minus g and g, all right? And g is a function of the T cell population, okay? And that tells you when you switch from growth to decay and then back. So here's how that works. So think of it this way. So this is my function g. Just take an example. Let's just take a fixed value of the immunogenicity function. Just take it as fixed. Let's say it's 0.1. Well, then this g function is very steep and flattens out as a function of n. That means that it's very, very sensitive to changes in the immune system, okay? So if the I of t is very small, it very quickly, as the immune system rises, it very quickly goes from a growth phase to a regression phase. On the other hand, if I of t is larger, it much more slowly goes from a growth to a regression. So it's more, so it's less responsive to the immune system, all right? So that's going to be a key for us, that function g. And it's a function, as I said, of n of t, all right? So you're basically switching from dominance by AG to dominance by AR, and then back again to dominance by AG, and so forth and so on, as the immune system goes up and down. And whether or not the immune system goes up and down is controlled by this H function, which is either positive or negative. Yeah?
I have to ask something really evil.
Uh-oh.
You qualitatively could get the same by just having two different phases. In one is kind of you favor cells of type X1, and the other phase you favor type X2. So you don't really need a game to get this qualitatively. So what's the evidence that you actually need something like the Prisoner's Dilemma?
Well, I'm not saying that this is the only model that will do this.
But,
You don't.
but I would be very happy if there would be evidence that you actually have a Prisoner's Dilemma in this growth phase, and not just a growth advantage of the, say, kind of X2 or defector cells.
I mean, I think of Prisoner's Dilemma as a generic way of modeling a system that has a fitness gap between the competitors.
Okay.
So I'm not claiming that I have experimental papers that I've seen that So Prisoner's Dilemma payoff matrix is one of the fairly, I mean, you know this as well as I do, one of the fairly standard ways of modeling, let's say a Gompertzian or logistic kind of a growth.
You would also get logistic growth if, for example, the entries in the first line of this AG matrix would just be 1, and then the second line would be R. Just have some growth advantage. You also get the same logistic growth, or actually.
Oh, I see what you're saying. That's true. That's true. Yeah,
You have fewer parameters, and that's why people always tell me I should work in games.
Yeah.
So I'm trying to tell you why I should get.
Well, okay, I'm not going to belabor the point because there's obviously many models that can do the same thing. What can I say? All right. All right. So let me just show you what the model then, one of the things, the starting point of the model. And again, I haven't yet talked about the control functions. So if the parameters are right, then you get a balance of these things and you get a fundamental cycle. So what I'm showing here is the oscillations of the X2 variable, so the cancer cell frequency, and the T cell population, all right? And the thing is, they're out of phase, they're slightly out of phase, and their amplitudes are different. And that's more or less how it should be. We understand totally how the phase difference between the two and the amplitude difference between the two scales with all of our parameters in the model, and so that allows us to tune things to different data, and I'll show a picture in a second. So that is an example of the fundamental cycle in action for this system. And the gray areas here, this is, as I said, we're running this thing stochastically by having the coefficients in the deterministic model vary stochastically over a certain range, and that gives us our uncertainty in these things. And typically we take plus or minus 5% around some baseline values.
Sure.
Yeah.
Very naive question as well. Is there any biological system that resembles that dynamic in the sense that you really have like I mean, there's some tumors that have like cyclic behaviors?
Here we go. Were you a referee on this paper? Because they made me dig out. So yes, that was, so I did find this, and the thing is, it's very difficult to get good data that not only tracks the volume of a tumor, which is obviously there's tons of papers on that, and at the same time tracks the immune system response in the same person or mouse, because you need to know the phasing. Well, here, 1994, nice paper. This is a mouse data, and it's they put a tumor in a mouse, and they don't give it chemo or immunotherapy, but they look at the immune response, and this would be an example of one part of a cycle of how the immune response, how the tumor grows, and then just the immune system kills the tumor volume. And so we can easily cook up the parameters in this model to match that.
They measure T cell populations along.
They measure the growth and decay of the tumor. Yeah.
But there is no so could
This is the volume of the tumor. This is, yeah, you're right. I don't have a corresponding oscillation of the T cells.
I think There's actually one scheme in leukemia which they measured in these collie dogs. You probably I like to look it up. There's a CML case where actually you have like a type of a oscillation in leukemia, but I'm also not sure whether this is like triggered by the immune system. But it's very standard example. I think Mike Mackey did it back in the 90s.
Yeah. All right. So let me say a couple more things. So this would be this is what the model spits out in terms of the volume of the tumor volume. And I'm just showing it for different discrepancy parameters that basically, allows us to sweep through different kinds of growth curves. And as I say, this regression is due to the immune response, and the growth is due to the growth. So far, absolutely no chemotherapy, no immunotherapy. And there's a nice little...
Sure. Why does the tumor keep going higher and higher in the oscillations while the immune response does the same thing?
Because so the volume goes higher and higher. Is that what you're asking?
Yes.
That's because of this discrepancy parameter. And that's because the immune system doesn't have the ability to kill the entire tumor. So as you go through one phase, the growth phase is stronger than the ability for the immune system to kill it. If it was equal, we wouldn't need immunotherapy or chemotherapy. So again, you can tune that and to data. All right, so there's also a nice formula for the volume as a function of the period, which is nice. All right, so let's go on. Let me focus on some questions that we can address in this model, which are interesting, and so I'm going to walk you through some of the results. So the questions we address would be these. Do chemotherapy and immunotherapy schedules commute? That is something that's not well understood. In other words, if you give a pulse of chemotherapy and then you give a pulse of immunotherapy, is the response the same as if you switch the order and you first give the immunotherapy and then you give the chemo? So we first look at that. Is it better to combine chemotherapy and immunotherapy dose schedules sequentially, one after the other, or concomitantly at the same time? When is the optimal time to start and stop immunotherapy treatment? Very, very, very unknown. What is the benefit of using more complicated time-varying dosing schedules compared to pulse dosing protocols, which is the typical thing? So I'll walk you through some of that. Some nice papers that we learned a lot from was this Nature paper on dose combinations for immunotherapy, and this one, which I'm going to pull a figure out in a minute. What, why, where, and when, bringing timing to immuno-oncology. Really, really nice review paper on what is known about timing immuno and chemotherapy with the cancer immunity cycle. All right, so now the final part, 3 of the model, is the control theory part, where we talk about C of t and theta of t. So, as Ruth asked, our chemotherapy function, C of t, it comes up in this form, let's say of the fitness F2. And this, when C is 0, there's no selection pressure on the cancer cell population. And when C is positive, it applies selection pressure on the cancer cell population. All right, so that's how we implement chemotherapy. And in general, C is going to be for us a function that's always between 0 and 1. And theta, as I said, is our immunotherapy parameter, time-dependent in general. Because of the idiosyncrasies of the model, it's always between 2 and 5. So when theta is 2, immunotherapy is off, and it's just a natural immune response. And when you increase theta, then you're lowering the activation threshold, and that then indirectly affects the selection pressure on the cancer cells. All right? Okay, so let me walk you through a couple of things. So we can shape pulse dosing in all kinds of ways, and so we use a certain that function for C of t, and that allows us to basically construct our pulse doses, how much chemo we give, how long, and so forth, when we start, when we stop. And what we're doing is we are always taking rational multiples of the fundamental cycle. And so we tested basically M and N for different values, relatively small because, there's not really much difference between, you want to look at 1/4, 1/3, 1/2, and things like that. You don't really care about taking M over N to be 109 over 107. So multiples of the cycle, of the fundamental cycle. And so let me just show you a quick first result, and that is this. So just chemotherapy. If you start, so let's take a quarter pulse, and we're comparing now, starting at the peak of the cancer cell population as compared to starting at the trough. And the question is, which is better in terms of the response? Well, clearly, starting at the trough is better because of this. Sorry, starting at the peak is better because it has more of an effect after you're done with that pulse, all right? So things like that will be the kinds of tests we've done, thousands of different kinds of tests. And as you can imagine, there's lots of different combinations and tests you can do. So let me now keep going. For the immunopulse dosing, we also take a similar function, but we do something else with the immunopulses because you need a couple of things to make it more realistic. One is that the system doesn't respond immediately to a pulse of immunotherapy. There's a little bit of a lag time. And also, when you stop the immunotherapy, again, the system doesn't immediately respond. There's a little bit of a lag time with that. And so we can shape the profile, with this function. And that's what we do to model, let's call it the delay in the immune system response to immunotherapy and the immune system memory effect. Again, there are other ways of modeling this. You could do a delay equation, but that makes it a little bit awkward. All right, so let's look at a simple example here. So now we're just doing one pulse of immunotherapy. Here I'm comparing 1/4 pulse versus 1/2 pulse. The quarter pulse is at the peak. The half pulse is at the trough. And again, you can see that the half pulse at the trough works much better.
Sorry, do you know what happens next? Like, this shows one cycle after the therapy.
Oh, what happens over longer times? Yeah, I do know what happens next. It basically is not perfectly periodic.
And this one is worse, right?
Yes.
Continues to be worse than that.
Yes, it continues to be worse. But keep in mind that these studies are basically short time studies. I will show something a little bit longer towards the end. But these are responses relatively short time. But yes, we certainly did look longer, and this never really got better at all. All right, so the first question that I addressed was the question of whether sequential pulse therapies commute or not, and the answer is they do not commute. So I'm showing here first an immunotherapy pulse, then a chemotherapy pulse, and then I'm switching the order. And they definitely have different responses because of that, the order in which you do it. Same dose pulse. And it's sort of clear why they don't commute, and that's because the system response to the first immunopulse is different from the first chemopulse. So whichever one you give first, the system responds to it, and then you're applying the second pulse to a different system in each case. So, yeah.
Quick question, comment. I mean, there's a notion out there that chemo can help an immune response because you blow up the tumor cells and it releases antigen. I'm not sure to what extent this is actually true or not, but I've heard it many times.
Say it again.
If you give chemo first, it destroys the tumor cells. That releases tumor antigens, which is beneficial for the immune response. If that was true, then giving chemo first could be beneficial.
Yeah, I'm not sure, but that is basically the cancer immunity cycle would sort of say that because the antigen release is related to the death of the cancer cells, and that does trigger the immune response. So yeah, I don't know what to say about that. Okay. Concomitant pulse therapy, again, we went through different possibilities there. We went through possibilities where they're, well, concomitant, they're given at the same time, starting at the same time. The top one shows exactly the same pulses, and the bottom one shows that the immunotherapy pulse is twice as long as the chemotherapy pulse. And as you can see, the one that works the best is that you give a quarter pulse of chemotherapy and a half pulse of immunotherapy, and you start them both at the trough. And I'm just taking a little small selection of figures of many that we tried and showing you kind of the highlights. And so this is all just little snapshots of lessons that we can learn from little pulses and combinations. So then we asked the question, well, suppose you didn't do pulse therapies, suppose you did an optimization problem and you allowed a theta of t and c of t basically to be a time varying, and you just optimized, and you asked what would the optimization produce? And so interestingly, so we did that, and basically it's more or less robust to this idea that the immunotherapy should start first, and it should basically be twice as long as the chemotherapy. So we did the optimization based on a cost function. We have a running cost through the full time, and then we have an end cost where we're trying to minimize the tumor volume at the end of the time. And so this would be an example of a result of optimized time-dependent chemotherapy, not pulse therapy. But this is what the time-dependent therapies end up looking like. The immunotherapy, the chemotherapy. More or less, immunotherapy twice as long as the chemotherapy. All right.
Is that analytical?
No, this is numerically.
Numerical optimization?
Yes. Yeah, I don't know how to do that analytically. No, too hard. Yeah. Okay, so I'm running out of time. I am going to just quickly show you that it's interesting and useful to do a sensitivity analysis on this model, which we did the Sobel analysis, which basically is a variance-based analysis where based on the variances associated with the spread of the distribution of each of the parameters in the model, you want to know what the variance is in the outputs of the model. And of course, those are going to be complicated because they're not just going to add up linearly. And so the Sobel method basically lays this out for you. So here I'm showing the sensitivity of the cancer cells, the immune response, and the tumor volume for the first order Sobel indices, meaning each of the variables are varied independently. And then I'm showing the second order, meaning when you combine the variance associated with two of the parameters, then how does that change? So the upshot here is that for the cancer cells, the system is most sensitive to epsilon, which was the immune response timescale, how quickly the immune system responds to changes in the cancer cell population, and G1, which was basically the growth phase of the payoff matrix. And anyway, there's a lot to say about sensitivity analysis, but it's interesting and worthwhile, I think. So I'm wrapping it up here. A couple more mechanisms that the model builds in, 3 additional important biological mechanisms of the model. If you do long time, then you really should probably take into account chemo resistance over longer times. And so the way we do that in this particular model is our chemotherapy function, the effective chemotherapy, then decays exponentially, which is one simple way of saying that the tumor, the cells become more resistant to the chemotherapy over long times. And so that's an immune escape mechanism. That's one of the many known immune escape mechanisms. And here it would be an example of this. This is the cancer cell population, function of longer times. The tumor shrinks because of the chemotherapy, and then it becomes resistant and regrows. As I said, there's also this tumor immunogenicity function allowing for immune evasion as the tumor matures, and this would be an example of that. The immune system starts killing the cancer cells, and then eventually the tumor becomes more resistant to the immune system, and it's also one of the known escape mechanisms. And finally, the immune system is suppressed, and so the model shows that as well. And that's this S term in the model that I talked about. All right. So as I said, I want to show, so this is taken from this nice paper that I got a lot of the ideas from this model on. They weren't doing modeling, they were basically talking about these questions. So this group at MIT, Dane Woodruff, I think, is the leader of this, this particular study. They wrote a nice review paper, What, Why, Where, and When, bringing timing to immuno-oncology, and in 2018, and they reviewed what they knew and basically the clinical trials that were in place, which basically did timing studies based on the tumor immune cycle, different kinds of drugs at different phases in this cycle, and how the tumor responds to that timing. And absolutely, there's evidence that taking advantage of the timing of different drugs during different phases of this cycle is definitely beneficial. I'm not saying that it's easy, because it is difficult to track exactly where you are in a cycle and things like that. So there's certainly challenges, but it is beneficial to do that. And so, I think that there's a lot more to learn from these kinds of models on the kinds of things that are possible. Okay, so let me wrap it up and just say that there's some important model ingredients here that we have in here that are important. One is it's based on cancer cell, healthy cell replicator dynamics. It's a cyclic process. There's stochastic parameter variation. There's immune evasion mechanisms. There's immune suppression mechanisms. There's an immune activation threshold. There's immunotherapy delay response. There's immunotherapy memory response. There's chemoresistance mechanisms. There's chemotherapy and immunotherapy time-dependent control knobs in the model. We looked at single pulse and multi-pulse dosing schedules, and then we did optimized dosing schedules. So that basically is where we are in this model. And there is definitely a lot more to do, particularly, I would say, the stochastic aspect of this model, the finite cell versions of this model and things like that, which are all very interesting. So let me just say this. Conclusion slide would be lots more to learn from this model. So yeah, thanks very much.